Hello, my dear students. Welcome to Sankalp Learning Classes. So, already we have started the class on the chapter Sexual Reproduction in Flowering Plants. So, today we are going to continue the same chapter. Okay. Yes, in the last class, what we have studied? So, we have studied about the microsporogenesis, how the male gametes are formed, how the male gametophyte is formed, and about the female reproductive structure and the formation of the female gamete. So, megasporogenesis. So, that also we have studied in the last class. So, now in this class, we are going to continue the chapter. So, the today's first topic is pollination. Okay. What is pollination? So pollination is the mechanism to achieve this objective. So the transfer of pollen grains to the stigma of a pistil is termed as a pollination. Pollen grains, so that is the male gametophyte, that yellow color structure alva, yellow color structure in pollen grains formation agide male gametophyte alva. So adenagbeku stigma ge transfer agbeku. For example, just imagine this is the stigma. Okay, so stigma, style, ovary. So the pollen grains are here. Just imagine this is the stamen and pollen grains are here. So this transfer of the pollen grains to the stigma of the flower, so that is called as a pollination. The pollen grain that is maybe from the another plant also. Okay, but the what is the meaning of the pollination? The transfer of the pollen grains to the stigma of the pistil. So this is the stigma of the pistil, that is termed as pollination. Then there are different kinds of pollination. Let us study about the kinds of pollination. First one, autogamy. Here, what is autogamy? Here, transfer of the pollen grains from the anther to the stigma of the same flower. So here, just imagine this is the stigma, style and ovary. So just imagine this is the flower. Okay. Just roughly I'm drawing this. Okay. So this is the flower. Here, stamens are there. So pollen grains, they are transferring to the stigma of the same flower. So within a flower, so just know the meaning, transfer of the pollen grains from the anther to the stigma of the same flower, that is known as autogamy. Here in a normal flower which opens and exposes the anthers and stigma completely, autogamy is rather rare. Here, autogamy is in such flowers requires synchrony in pollen release and stigma receptive, and also the anthers and stigma should be like close to each other for the cell pollination can occur. Here, what is the meaning of autogamy? Such flowers require synchrony in pollen release and the stigma receptive. Here, so in this type of pollination, the stigma should be receptive. The stigma should be ready for the uh, reception of that pollen grains, and pollen grains should release at that time only. So in that particular situation only the pollination occur. Enagirbeku illi pollen grains ready irbeku and stigma also should be ready alva. So stigma matte pollen grains eradu ready iddaga matra enagatte illi pollination agatte. Okay, and they should lie close to each other. So ಎರಡು ಏನಾಗಿರ್ಬೇಕು, they should lie close to each other. So in that case only the autogamy can occur. Okay, so that is the meaning of the autogamy. Then some plants such as viola and oxalis and commelina produces two types of the flower. So remember the examples ಅನ್ನ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. Viola, oxalis and commelina produces two types of the flowers. What are the two types of the flowers? Here, Chasmogamous flowers. What is the meaning of chasmogamous flowers? Which are similar to flowers of other species with exposed anthers and stigma. Here, the flowers, here you can observe the chasmogamous flowers means which has the exposed anther and stigma. So what you observe in hibiscus. So in hibiscus, you can observe the exposed stigma and the anthers, right? But clistogamous flowers which do not open at all. In such flowers, the anthers and stigma lie close to each other. So the flowers, they do not open at all. Just to observe these flowers, clistogamous flowers, you can observe here. Clistogamous flowers means they will not open at all. They will close, remain close. So in that flowers, the stigma and the pollen, the stigma and the male reproductive structure, they lies anthers, so they lie close to each other. So in clistogamous flowers, so that is invariably autogamous because there is no chance of cross pollination or landing of the another pollen grain on the stigma. So ಇಲ್ಲಿ ಈ clistogamous flowers ಏನಾಗಿರುತ್ತೆ ಅಂದ್ರೆ ಅಲ್ಲಿ ಅಟೋಗ್ಯಾಮಿ ಆಗಿರುತ್ತೆ ಅಲ್ವಾ? ಯಾಕೆ ಅದು ಓಪನ್ ಆಗೋದಿಲ್ಲ ಅಂದ್ರೆ ಪಾಲಿನೇಷನ್ ಅದೇ ಫ್ಲವರ್ ಅಲ್ಲಿ ಆಗ್ಬೇಕು ಅಲ್ವಾ? ಸೊ ದಿಸ್ ಕ್ಲಿಸ್ಟೋಗ್ಯಾಮಸ್ ಫ್ಲವರ್ಸ್ ಆರ್ ಇನ್ವೇರಿಯಬಲಿ ಅಟೋಗ್ಯಾಮಸ್ ಅಂಡ್ ಕ್ಲಿಸ್ಟೋಗ್ಯಾಮಸ್ ಫ್ಲವರ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಅಸ್ಯೂರ್ seed set even in the absence of the pollinators. So ನಿಮಗೆ ಇದನ್ನ MCQ ದಲ್ಲಿ ಕೇಳಬಹುದು. So in the absence of the pollinators, so these flowers they assure the seed set even in the absence of pollinators. Pollinators ಬೇಕು ಅಂತ ಇಲ್ಲ ಅಲ್ವಾ? So in the absence of pollinators, they are able to perform the pollination. So that is the clistogamous flowers. Okay, so please know the meaning of chasmogamous flowers and clistogamous flowers. And so what happens in the clistogamous flowers and the chasmogamous flowers? Next one, Geitonogamy. Geitonogamy means, so you know the meaning of autogamy. Geitonogamy means transfer of pollen grains from the anther to the stigma of the another flower of the same plant. Just imagine, this is the plant. So there is a flower. Okay, so this is a flower and this is the another flower. Just imagine. So these are the flowers. Okay. So here in this plant, so autogamy means within the same flower, the transfer of the pollen grains takes place within the same flower, stigma of the same flower. But here, transfer of the pollen grains, just imagine the pollen grains are here. Pollen grains are in this flower and this transfer of the pollen grains to the stigma of the another flower. So this is the another flower but in the same plant. Stigma of the another flower of the same plant. So I hope you get the meaning of this. So ಅರ್ಥ ಆಗಿಲ್ಲ ಅಂದ್ರೆ ನೀವು ಕಮೆಂಟ್ ಮಾಡಬಹುದು. Pollen grains, they transfer to the stigma of the another flower of the same plant, that is called as the Geitonogamy. Although Geitonogamy is functionally cross-pollination, so illenagatte cross-pollination agatte, but involving the pollinating agents, genetically it is similar to autogamy since the pollen grains come from the same plant. Here the pollinating agents are required. Illi pollen grains ಈ ಫ್ಲವರ್ ಇಂದ ಇನ್ನೊಂದು ಫ್ಲವರ್ ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗಬೇಕು ಅಂದ್ರೆ ಅಲ್ಲೇನ್ ಬೇಕು? ಪಾಲಿನೇಟಿಂಗ್ ಏಜೆಂಟ್ಸ್ ಬೇಕು ಅಲ್ವಾ? Then it is invariably, so that is genetically autogamy. Genetically same plant ಅಲ್ಲಿ pollination ಆಗಿರೋದಕ್ಕೆ, so we can say that autogamy, but the pollinating agents are required here, that is the Geitonogamy. Okay, so ನಿಮಗೆ ಡಿಫರೆನ್ಸ್ ಕೇಳಿಸ್ತಾರೆ, Geitonogamy and autogamy or Geitonogamy and chasmogamous flower. So ಈ ತರ ನಿಮಗೆ ಡಿಫರೆನ್ಸ್ ಅನ್ನ ಕೇಳ್ತಾರೆ. Please know the meaning of these all type of pollination. Okay, then one more type of the pollination, that is the Xenogamy. Transfer of the pollen grains from the anther to the stigma of the different plant. So this pollination is known as a xenogamy. So just imagine, so there is a flower. Okay. So just imagine this is the flower, and there are pollen grains from this plant, this flower to the, so here stigma. Here the pollen grains, they transfer to the stigma of the another flower of the another plant, not the same plant. So same plant ಆಗಿದ್ರೆ ಏನಾಗ್ತಿತ್ತು, Geitonogamy ಆಗ್ತಿತ್ತು ಅಲ್ವಾ? But here the pollen grains, they transfer to the stigma of the different flower of the different plant. That is called as xenogamy. So xenogamy that occurs in papaya, so which produces the ಯುನಿಸೆಕ್ಸುವಲ್ ಫ್ಲವರ್ಸ್. ಸೊ ಫ್ಲವರ್ಸ್ ಅಲ್ಲಿ ಏನಿರುತ್ತೆ, ಸೊ ಯುನಿಸೆಕ್ಸುವಲ್ ಫ್ಲವರ್ಸ್ ಅಂದ್ರೆ ಒಂದು ಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಸ್ಟ್ರಕ್ಚರ್ ಇರಬಹುದು ಅಥವಾ ಫಿಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಸ್ಟ್ರಕ್ಚರ್ ಇರುತ್ತೆ ಅಲ್ವಾ. ಸೊ ಆ ತರ ಒಂದು ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ, ಸೊ ದಿಸ್ ಟೈಪ್ ಆಫ್ ಪಾಲಿನೇಷನ್ ಅಕರ್. ಸೊ ಯು ಕ್ಯಾನ್ ಗಿವ್ ದ ಎಕ್ಸಾಂಪಲ್, ದಟ್ ಈಸ್ ಪಪಾಯ. This is the only type of pollination which during the pollination brings genetically different types of the pollen grains to the stigma. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, genetically different ಇರುವಂತ, ಇಲ್ಲಿ ಬೇರೆ ಬೇರೆ plants ಮಧ್ಯ pollination ಆಗುತ್ತೆ ಅಲ್ವಾ. So where the genetically different types of pollen grains to the stigma is transferred. So that is xenogamy. So very important, autogamy. And so in autogamy, chasmogamous flowers and the clistogamous flowers, then Geitonogamy, Xenogamy, these all are very important for your board as well as CET exam. Okay, so please remember the meaning, examples. So that is very important. So here for the pollination, pollinating agents are required, right? So here agents of pollination means sorry, means to transfer of the pollen grains. So there are some agents are required. Plants use two types of the agents, that is abiotic factors or the biotic factors. Abiotic means wind and water, and the biotic means animals. So these two agents are used to achieve the pollination. Okay, so majority of plants use the biotic agents for pollination. Only small proportion of plants use the abiotic agents. Okay, so let us see the abiotic agents, so which are used for the pollination. First one is the pollination by the wind. So here wind is the abiotic agent which is used to transfer the pollen grains to the stigma of the flower. Okay, so here the wind is used. Wind means that is the that is the abiotic factor. Okay, so it is pollination by wind, which is more common among the abiotic pollinators. Pollination. Wind pollination also requires that the pollen grains are light and non-sticky, so they can transport it in the wind current. So this wind pollinated plants means the pollen grains that are transferred through the wind. So these plants have some characteristic features. So they will ask, write the characteristic features of the wind pollinated plants. Okay, so wind pollinated plants characters bariri anta keltare, or some characters are given. So in multiple choice question also, multiple choice question allu kuda keltare. Wind pollination also requires the pollen grains are light and non-sticky, so they can be transported in the wind current. So ಈ ವಿಂಡ್ ಅನ್ನ ಯೂಸ್ ಪಾಲಿನೇಷನ್ ಬೈ ವಿಂಡ್ ಅಂದ್ರೆ ಏನಾಗುತ್ತೆ, ಆ ವಿಂಡ್ ಅಲ್ಲಿ ಏನಾಗಬೇಕು, ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಕ್ಯಾರಿ ಆಗಬೇಕು ಅಲ್ವಾ. ಇಫ್ ದ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಆರ್ ಹೆವಿ ವಿಚ್ ಇಸ್ ನಾಟ್ ಕ್ಯಾರಿಡ್ ಬೈ ದ ವಿಂಡ್, ಸೊ ದ ಪೋಲನ್ ಗ್ರೇನ್ ಶುಡ್ ಬಿ ಲೈಟ್ ಅಂಡ್ ನಾನ್ ಸ್ಟಿಕ್ಕಿ. They often possess well exposed stamens so that the pollens are easily dispersed into the wind currents. So they have the well exposed stamens. You can see the well exposed stamens. So stamens exposed ಆಗಿದ್ರೆ ಏನಾಗುತ್ತೆ, ಸ್ಟೇಮನ್ಸ್ ಮೇಲೆ ಏನಿರುತ್ತೆ, ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಇರುತ್ತೆ ಅಲ್ವಾ. ಈ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಏನಾಗುತ್ತೆ, ಈಸಿಲಿ ವಿಂಡ್ ಥ್ರೂ ಕ್ಯಾರಿ ಆಗುತ್ತೆ. ಸೊ ದೇ ಹ್ಯಾವ್ ದ well exposed stamens so that the pollens are very easily dispersed into the wind currents, and large often feathery stigma to easily trap the airborne pollen grains. So ಇಲ್ಲಿ ಸ್ಟಿಗ್ಮಾ ಯಾವ ತರ ಇರುತ್ತೆ, ಲಾರ್ಜ್ ಸ್ಟಿಗ್ಮಾ and that easily trap the airborne pollen grains. ಸ್ಟಿಗ್ಮಾ ಏನಾಗಿರ್ಬೇಕು, ಲಾರ್ಜ್ ಆಗಿರುತ್ತೆ, so that is able to trap the pollen grains, and well exposed stamens. So that is the characteristic. And wind pollinated flowers often have single ovule in each ovary and numerous flowers packed into a inflorescence. So that is also one of the important character. A familiar example is corn cob. The tassels you see are nothing but the stigma and the style. So idanna nimge multiple choice alli keltare. The corn cob, the tassels you see, that is ಸ್ಟಿಗ್ಮಾ ಅಂಡ್ ದ ಸ್ಟೈಲ್. ಕಾರ್ನ್ ಕಾಬ್ ಮೀನ್ಸ್ ನೀವು ಈ ಕಾರ್ನ್ ಅಲ್ಲಿ ನೋಡಿರ್ತೀರಲ್ವಾ ಈ ತರ. ಸೊ ಟ್ಯಾಸೆಲ್ಸ್, ಸೊ ದೇ ಆರ್ ನಥಿಂಗ್ ಬಟ್ ದ ಟ್ಯಾಸೆಲ್ಸ್, ಈ ರೀತಿ ಇರುತ್ತೆ ಅಲ್ವಾ. ಜಸ್ಟ್ ಅ ಮಿನಿಟ್. ಇಲ್ಲಿ ಸೊ ಕಾರ್ನ್ ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಈ ರೀತಿಯಾಗಿರುತ್ತೆ ಅಲ್ವಾ. ಸೊ ದಟ್ ಈಸ್ ದ ಟಸೆಲ್ಸ್. ಟಸೆಲ್ಸ್ ದಾರದಂತ ಒಂದು ಸ್ಟ್ರಕ್ಚರ್ ಅನ್ನ ನೋಡಿರ್ತಿರಲ್ವಾ. ಸೊ ದಟ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ ದ ಸ್ಟಿಗ್ಮಾ ಅಂಡ್ ದ ಸ್ಟೈಲ್ ವಿಚ್ ವೇವ್ ಇನ್ ದ ವಿಂಡ್ ಟು ಟ್ರ್ಯಾಪ್ ದ pollen grains. So adenadu, so that is stigma and the style which is used to trap the pollen grains which is present in the wind. Wind pollination quite common in grasses. Okay, so idannu kuda MCQ dalli keltare, wind pollination common agi elli agutte anta. So that is the important question. And here main agi keluvanthaddu nimge characteristics, wind pollinated plants characteristic anna keltare. So you have to write these all very important characters so for in your exam. Okay, then second type of the abiotic agent, that is water. Pollination by water is quite rare in the flowering plants and it is limited to about 30 genera, mostly in monocotyledons. Very important. So, pollination by water. So, water pollinated flowers ಏನಿರುತ್ತೆ, so they are very rare. So, limited up to 30 genera, mostly monocotyledons, monocot plant ali. So, it is observed. And water is regular mode of transport. Here, the water is used for the transfer of the pollen grains. Okay, for the male gametes among the lower plants groups such as algae, bryophytes, and pteridophytes. So, water is the common or regular mode of transport for the gametes in case of algae, bryophytes, and pteridophytes. But what we are studying about the angiosperms, right? Here, it is believed, particularly some bryophytes, pteridophytes, that their distribution is limited because of need of water for the transport of the male gametes and the fertilization. Here, some bryophytes and pteridophytes, they are limited to the particular region, right? So, what is the reason behind that? Because they need water for that distribution of the male gametes. So, that is the reason. And here, some examples for water pollinated plants are Vallisneria, very important, and Hydrilla, which grow in fresh water, and several marine sea grasses such as Zostera. Okay, so example for the marine sea grass, that is Zostera, and Vallisneria and Hydrilla, they are the freshwater plants and which are pollinated through the water. Not all aquatic plants use for pollination, water for pollination. So if the plant is growing in water, so that plant is not using the, all plants are not using the water for their pollination. For example, water hyacinth and water lily. So the flowers emerge above the level of water and are pollinated by the insect or wind as the most of the land plants. So here some plants which are growing in the water, but they are not pollinated by the water, they are not using the water for their pollination. Just imagine, so this is the water level. Okay, so some plants like Vallisneria, Hydrilla, Zostera, they use the water for the transfer of the pollen grains. But some plants like water lily, water hyacinth, so they grow, just imagine, so this is the water lily, so that is growing above the level of water and which is having the stigma. Just imagine, this is the stigma and the flowers above the water level, and which is these flowers are pollinated by wind or by the insect. Okay, so which is growing in water but which are using the other agents like wind and the insects for their pollination. So the pollen grains are not transferred through the water, they are transferred through the, they are using the some other methods. Okay, so here in Vallisneria, how the pollination takes place, just see the diagram. In Vallisneria, the female flower reach the surface of the water by long stalk, and male flowers or the pollen grains are released on the surface to the water. Okay, here, here the Vallisneria, the female flowers reach the surface of the water. Just imagine, this is the surface of the water and this is the female flower. Okay, the female flower that reach the surface of the water. Then the male gametophyte, that is pollen grains, they are released at the surface of the water. Okay, just imagine, this is the Vallisneria flower. So that is reaching the surface of the water, and the male flowers or the pollen grains, so they are released on the surface of the water. So these are the pollen grains. So these pollen grains now reach the stigma of this flower. So like this the pollination occur in the Vallisneria, and these pollen grains are carried passively by water. Some of them eventually reach the female flowers and the stigma. So then another group of plants, they are pollinated by, for example, sea grasses. The female flowers remain submerged. So in which plants, so in which water pollinated plants, the female flowers, so that reach the surface of the water, that is in the Vallisneria. But in sea grasses, the female flower that is submerged. So you can see the female flower, so that is submerged in water, so which is present inside the water, that is in the sea grasses. Okay, Zostera. Here female flowers remain submerged in water and the pollen grains are released inside the water, and pollen grains in many such species are long ribbon like, and they are carried passively inside the water. So the male flowers, they release the pollen grains, and these pollen grains, what are the characteristics of the pollen grains of the water pollinated plants? So they may be long ribbon like and they are carried passively inside the water. Some of them reach the stigma of the flower and achieve the pollination. So, and in most of the water pollinated species, pollen grains are protected from wetting by mucilaginous cover. Okay, so that is the main thing. Then pollination by insects. How the pollination takes place by insects? So insects, majority of the plants, so they are pollinated by the insects or animals like bees, butterflies, not only insects, mainly bees, butterflies and beetles, wasps, ants, moths and birds such as sunbirds and hummingbirds, and bats are the most common pollinating agents. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, insects or animals through ಏನಾಗುತ್ತೆ, pollen grains, so that is transferred to the stigma of the flower. So even larger animals like some primates, lemurs ಇರಬಹುದು ಅಥವಾ tree dwelling animals irbodu or reptiles like garden lizard, gecko. So these also achieve so they are also act as a pollinators, not only insects, some animals also. Here so these insect pollinated plants, they are colorful, fragrant and rich in nectar. So water pollinated plants irbodu, wind pollinated plants irbodu, they are not fragrant, colorful or rich in nectar, because they do not, they are, there is no need to attract the some insects or anything. But here the majority of insect pollinated plants, they are colorful. So colorful petals irutte and fragrance irutte, nectar irutte alva. So that is the main characteristic of the insect pollinated plants. Insect anna attract ಮಾಡಬೇಕು ಅಲ್ವಾ, ನೆಕ್ಟರ್ ಇರಬಹುದು ಅಥವಾ ಫ್ರಾಗ್ರೆನ್ಸ್ ಇರಬಹುದು. ಸೊ ಅದರಿಂದ ಅಟ್ರಾಕ್ಟ್ ಮಾಡಬೇಕು. ಸೊ ದೇ ಹ್ಯಾವ್ ದ these type of characteristics. When the flowers are small, number of flowers are clustered into inflorescence to make them conspicuous. Animals are attracted to the flowers by colors and or the fragrance. So that I explain. And the flowers pollinated by flies and beetles secrete foul odors to attract these animals. To sustain the animal visits, the flowers have to provide the rewards to the animals. So these flowers, they provide the rewards to the animals by providing the nectar or pollen grain are usual floral rewards. The nectar, so that is also one of the important rewards. So that is given to the animals. For harvesting the rewards from the flower, the animal visitor comes in contact with the anther and the stigma. So sometimes the body of the animal gets coating of the pollen grains, which are generally sticky in animal pollinated flowers. So sometimes what happens, the pollen grains that is sticked on the body of the animals and that is transferred to the, when it is visited to the another flower, that is transferred to the stigma of the flower. Okay, that is the, so they will also ask the write the characteristics of the animal or insect pollinated flowers. In some species, floral rewards are providing safe places to the lay the eggs. An example is that the tallest flower, that is Amorphophallus, very important. So that is about the 6 feet height. So a similar relationship exists between the species of moth and the plant of yucca, where both species, moth and the plant, cannot complete their life cycle without each other. So ಕೆಲವೊಂದ್ಸಲ ಏನಾಗುತ್ತೆ, so the insects, they lay the eggs inside the flower. Okay, so they the flowers providing the safe place for the insect egg. So that is how the mutual relationship between the flower and the insect. The moth deposit here, the moth and the yucca plant, the moth deposits the eggs in the locule of the ovary of the flower, in turn gets pollinated by the moth. The larvae of the moth come out of the eggs as the seeds start developing. The moth lay the egg inside the plant, flowers of the plant, and the eggs that is present inside the locule of the ovary. So when the seeds start developing, the larvae that comes out from the flower. So that is how there is a mutual relationship between the moth and that plant. Okay, then outbreeding devices. So what is outbreeding devices? Here majority of the flowers produces the hermaphrodite flowers, means the male and female reproductive structure that is present in the same plant. Okay, sorry, same flower. So if they continue the self pollination, so one day plant ಅಲ್ಲಿ pollination ಆಗಿ ಏನಾಗುತ್ತೆ, there is some inbreeding depression. What is that inbreeding depression? Some genetically inbreeding depression, some diseases or some quality of or some nutrition, so that is lacking in that seeds or fruits. So that is the inbreeding depression. So ಒಂದೇ ಕಡೆ pollination ಆಗಿ ಏನಾಗುತ್ತೆ, so there is no genetical variability ಅಲ್ವಾ, so genetical variation ಇರೋದಿಲ್ಲ. So that may cause the inbreeding depression. So flowering plants have developed the many devices to discourage the self pollination. The flowering plants, they discourage the self pollination. Self pollination ಆದ್ರೆ ಏನಾಗುತ್ತೆ? So there is no genetic variation in that plant ಅಲ್ವಾ? So same ಇರುತ್ತೆ, generation to generation same ಇರುತ್ತೆ, and there is no adaptation for the changing environment ಅಲ್ವಾ? So to adopt that changing environment, they promote the cross pollination. Cross flower ಅಲ್ಲಿ cross pollination ಆಗಬೇಕು, self pollination ಆಗಬಾರ್ದು. Means the pollen grains that should transfer to the flower of the another plant. So that is the cross pollination. So here the plants develop some outbreeding devices. So what are these outbreeding devices? Nodi, flowers, the plants, they develop some outbreeding devices. So what are these? In some species, pollen release and sigma receptivity are not synchronized. So ಇಲ್ಲಿ ನೋಡಿ, ಕೆಲವೊಂದು ಫ್ಲವರ್ಸ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, pollen grains already ready, so they are mature, but stigma is not ready, stigma is not mature. So stigma is not ready to receive the pollen grains. So if the pollen grains transfer to the stigma, the pollination is not successful. So then what happens? When the pollen grains transfer to the another flower, then it will get pollinated. So this is very important here. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of the pollen. Pollen release agodkinta munche enagutte, stigma is ready, but the pollen grains are not ready. So in that way they achieve the cross pollination. In some other species, the anther and stigma are placed at the different positions so that the pollen cannot come in contact with the stigma of the same flower. So ಕೆಲವೊಂದ್ಸಲ ಏನಾಗುತ್ತೆ, the flower the stigma is, so that is long and height, but the pollen grains, they are covered, that is not able to transfer to the stigma of the same plant. So that is also another device. And the third device to prevent the inbreeding is self-incompatibility. This is the genetic mechanism and prevents the self-pollination. Means when the pollen grains that is transferred to the stigma of the same flower, the stigma that recognize it is the same pollen grain, so it is not able to germinate on the same stigma. That is the self incompatibility. Means when the same pollen grains transfer to the stigma, that is not able to germinate. Another device to prevent the self pollination is production of the unisexual flowers. Unisexual flowers means either male gametes, either male reproductive structure is present or female reproductive structure is present. So like that, which achieve the cross pollination. So ಇದನ್ನ ನಿಮಗೆ 5 mark question ಅಲ್ಲಿ ಕೇಳ್ತಾರೆ board exam ಅಲ್ಲಿ. If both male and female flowers are present on the same plant, such as castor and the maize, male and female flowers are present in the same plant, that is monoecious plants, it prevents the autogamy but not Geitonogamy. Autogamy is prevented, but Geitonogamy is not prevented. In several species such as papaya, the male and female flowers are present on the different plants. So that is the dioecious plant. Monoecious plants means within a same flower, male and female reproductive structures are present, that is monoecious, castor and maize. In papaya, that is the dioecious plant. Dioecious means male and female flowers are present in the different plant. So like that they achieve the cross pollination. So this is about the outbreeding devices. Let us study pollen-pistil interaction. What is pollen-pistil interaction? So here, when the pollen grain is transferred to the stigma, so pollen grain stigma transfer agide, so it will the pollen grain is able to germinate? No. Pollination does not guarantee the transfer of the right type of the pollen. So that is compatible pollen of the same species as the stigma. So sometimes the pollen grains is transferred to the stigma. So, but that is not the right type of the pollen grains. Pollen grains stigma transfer agide, but that is not the right type. So that is not the same species. ಬೇರೆ ಯಾವುದು species pollen grains stigma ಗೆ transfer ಆದ್ರೆ pollination ಆಗತ್ತಾ? No. Often pollen of the wrong type either from the other species or from the same plant also land on the stigma. So same species kuda land agbodu or from the different species also can land on the stigma. Okay, so the stigma is just imagine this is the stigma. So the stigma is able to germinate the right type of the that accept the right type of the pollen. The pistil accept the pollen and promotes the post-pollination events that leads to the fertilization. So here pollen-pistil interaction means when the right type of the pollen grain is landed on this stigma, then the further pollination, further things takes place. Post-fertilization, fertilization, then post-fertilization events takes place. So, ಬೇರೆ type pollen grains ಬಂದಿದ್ರೆ ಏನಾಗೋದಿಲ್ಲ, the stigma is not able to give the permission to germinate on that stigma. Okay, so that is called the pollen pistil interaction. If the pollen is not of the wrong type, the pistil rejects the pollen by preventing the pollen germination on the stigma or the pollen tube growth in the style. Here the stigma that is not allowed to germinate this pollen grain if it is a wrong type pollen grain. Wrong type pollen grain ಆಗಿದ್ರೆ ಏನಾಗೋದಿಲ್ಲ. So ಅದು germinate ಆಗ್ಲಿಕ್ಕೆ stigma that will not give the permission. Okay, so that is called as a pollen pistil interaction. Following the compatible pollination. So when the right type of the pollen grain that is landed on the stigma, that is called as a compatible pollination. Okay, compatible. The pollen grain germinates on the stigma and produces the pollen tube. Nodi, pollen, just imagine this is the pollen grain. So this is the stigma. Okay, so this pollen grain that forms the pollen tube inside this stuck. Okay, pollen tube ಈ ತರ formation ಆಗತ್ತೆ through one germ pores. The contents of the pollen grain move into the pollen tube and the pollen tube grows through the tissues of the stigma and style and reaches the ovary. So this pollen grain that is able to form the pollen tube through the germ pore. Germ pore alli sporopollenin absent irutte alva. Through that it produces the pollen tube. ಈ ತರ tube formation ಮಾಡುತ್ತೆ. Then it is able to reach the ovary. So just imagine this is the ovary. The pollen tube is formed like this. I will show the diagram. Yes, these are the pollen grains. When the compatible pollen grain, when it reaches the stigma, it is able to germinate and forms the pollen tube. ಇಲ್ಲಿ ನೋಡಿ pollen grain ನೋಡಿ pollen tube ಅನ್ನ formation ಮಾಡಿದೆ. That is able to reach the style, then able to reach the ovary. So that is mainly the female gametophyte. Embryo sac formation ಆಗಿದೆ ಅಲ್ವಾ? Inside the ovule there is a female gametophyte, embryo. So inside that, so it reaches the embryo sac. So the pollen grains are shed at two-celled condition, that is the vegetative cell and the generative cell. In such plants, generative cell divides and forms the two male gametes during the growth of the pollen tube in the stigma. So the generative cell that produces the two male gametes, very important, the generative cell during the formation of the pollen tube, it produces the two male gametes. Okay, then in the plants which shedded the pollen 3-cell stage condition, pollen tubes carries the two male gametes from the beginning. If the pollen grain shed at 3-cell stage, the pollen tube carrying the two male gametes from the beginning. And so when the pollen tube reaches the ovary, enters the ovule through the micropyle and enters the one of the synergids through the Filiform apparatus. So you have seen the diagram of the embryo sac. Your piliform apparatus will be there. They guide the these male gametes, and so like this, the male gametes, they enter the embryo sac. So this is how the pollen pistil interaction takes place. Next, artificial hybridization. So what is this artificial hybridization? So hybridization means where the pollen grains transfer to the stigma of the flower. So but artificial hybridization means, so mechanically, artificially, so this is the man-made process. So the to achieve the superior varieties, to produce the superior varieties, ನೀವು ಮಾರ್ಕೆಟ್ ಅಲ್ಲಿ ಕೇಳಿರಬಹುದು hybrid ಅಂತ ಅಲ್ವಾ. So that is having the superior characters, so in size or color or nutrient, anything to achieve the superior variety. So the breeders, so they will transfer the pollen grains to the stigma artificially. Okay, so that is the artificial hybridization process. So here, this is mainly achieved by the two process. That is, the first thing is the emasculation. So here in a flower, removal of the anthers, the male reproductive structure. So that is nothing but the emasculation. Artificial hybridization, that is achieved by the emasculation. Here, so this flower which is having the male and female reproductive structure. So here male reproductive structures are removed. This is the parent plant where the male reproductive structures are removed, and to this female reproductive structure pollen grains are transferred. So desired pollen grains, ನಮಗೆ ಯಾವ ಒಂದು pollen grains, ಯಾವ ಒಂದು characters ಬೇಕು ಅಲ್ವಾ, ಆ ಒಂದು characters ಇರುವಂತ plant ಇಂದ pollen grains ಅನ್ನ so they will transfer. So the first step is the emasculation. Okay, emasculation means removal of the anthers. That process is the emasculation. Emasculation ಆದ್ಮೇಲೆ ಏನ್ ಮಾಡ್ತಾರೆ, bagging. Bagging means the emasculated flowers, they are covered with a bag with a suitable size, generally made up of the butter paper, to prevent the contamination of the stigma with the unwanted pollen. So unwanted, ನಮಗೆ ಡಿಸೈರ್ ಇರುವ pollen grain ಮಾತ್ರ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಬೇಕು, ಬೇರೆ ಯಾವುದೋ pollen grains ಅಲ್ಲಿ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗಬಾರ್ದು. So the emasculated flowers, they are bagged. That process is called as the bagging. When the stigma is bagged, flower attains the receptivity. Mature pollen grains are collected from the anthers of the male parent, are dusted on the stigma, and the flowers are rebagged again, and the fruits are allowed to develop. So in the artificial hybridization ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ, so ನಮಗೆ ಡಿಸೈರ್ ಇರುವಂತ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಬೇಕು ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಅಂತ. So the pollen grains are dusted on the stigma. Okay, and it is again rebagged. So that is the process of the artificial hybridization. Then one more important topic, that is the double fertilization. So double fertilization, triple fusion. So in angiosperms, the double fertilization and the triple fusion process occur. So how it occurs? After entering to the synergids, so I will explain this process using one more diagram. Yes, this diagram. Okay, so here when the pollen tube growth occurs, the male gametes, two male gametes are released from one pollen grain. Okay, one pollen grain that releases the two male gametes. Okay, ಎರಡು male gametes ಇದೆ here within the embryo sac. Okay, so female gamete ilvali barbeku alva. So here in the micropylar region, synergids are there, egg cell is there. So this is the egg cell and central cell and the antipodals. Okay, so this is the structure of this embryo sac. The one male gamete which enter to the through the micropylar region and fuses with the egg cell. So that leads to the zygote. the one male gamete that fuses with the egg cell in the egg apparatus and that leads to the zygote. Male ಮತ್ತೆ female gamete ಏನಾಗಿದೆ fuse ಆಗಿದೆ, that leads to the zygote, and another male gamete is there ಅಲ್ವಾ, ಇನ್ನೊಂದು male gamete ಇದೆ, that male gamete that fuses with the central cell. So ಇಲ್ಲಿ already that is in a diploid condition because two nucleus already present, diploid condition. So one more male gamete enter and fuses with this and it will become triploid condition. So here the one male gamete fuses with the egg cell, that will become zygote, diploid condition, and one more gamete that fuses with the central cell, that is already in the diploid condition, 2n condition ಅಲ್ಲಿ ಇರುತ್ತೆ ಅಲ್ವಾ, and that will become 3n, triploid condition. So that is called as a double fertilization and the triple fusion. Yes. This result in the formation of the diploid cell, that is the zygote is formed, and another cell that forms the triploid condition, that is known as PEN, primary endosperm nucleus, that is always in a triploid condition. Primary endosperm nucleus that is always in a triploid condition. ಈಗ ಏನಾಗಿದೆ, the first fusion of the male and female gametes, that is known as a syngamy, and the fusion with the central cell that leads to the formation of the primary endosperm nucleus, that is PEN, that is triploid in condition. Yes, so this is the same. At this, the involves the fusion of three haploid nuclei, that is termed as triple fusion. Okay. Then the zygote that develop into an embryo and the primary endosperm cell that develops into an endosperm. Primary endosperm PEN that develop into an endosperm and zygote that develop into an embryo. So post-fertilization. So after the fertilization, so pre-fertilization events we have seen, that is microsporogenesis, megasporogenesis ಎಲ್ಲ ನೋಡಿದ್ವಿ ಅಲ್ವಾ. ಈಗ ಪೋಸ್ಟ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಯ್ತು, ಮೇಲ್ ಅಂಡ್ ಫಿಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಫ್ಯೂಷನ್ ಆಯ್ತು. ಸೊ ಆಫ್ಟರ್ ದ ಫ್ಯೂಷನ್ ಆಫ್ ದಿ ಮೇಲ್ ಅಂಡ್ ಫಿಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್, ವಾಟ್ ಆರ್ ದ ಇವೆಂಟ್ಸ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್, ವಾಟ್ ಆರ್ ದ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಫಾರ್ಮ್, that we have to study now. Okay, following the double fertilization, events of endosperm and embryo development and maturation of the ovule into seeds and ovary into fruit are collectively termed as post-fertilization events. So ovule that develops into seed and ovary that develops into fruit, and how the endosperm and embryo development takes place, these all are the post-fertilization events. Let us see the post-fertilization events one by one. What happens to the endosperm? Okay. Endosperm, that is nothing but that is formed by the fusion of the polar nuclei and the male gamete. PEN, primary endosperm nucleus. So the primary endosperm cell divides repeatedly and forms the triploid endosperm tissue. The cells of this tissue are filled with reserve food materials and used for the nutrition of the developing embryo. This PEN, so that contain the nutritive tissue that is used for the development of the embryo. Zygote ಏನಿದೆ ಅದು embryo ಆಗಿದೆ ಅಲ್ವಾ, embryo development ಗೆ endosperm is used. Okay. In most common type of endosperm development, the PEN, primary endosperm nucleus expansion ಅನ್ನ ಕೇಳ್ತಾರೆ, undergoes successive nuclear divisions to give rise to the free nuclei. This stage of endosperm development is called as a free nuclear endosperm. ಕೇಳ್ತಾರೆ free nuclear endosperm, what is that? And the number of free nuclei formed before the cellularization varies greatly. So ಇಲ್ಲಿ coconut water ಅಲ್ಲಿ ನೀವು ನೋಡಬಹುದು, the coconut water from the tender coconut, that is nothing but the free nuclear endosperm. Okay, and the surrounding white kernel, that is the cellular endosperm. So mainly the endosperm function is that is providing the nutrition for the developing embryo. Endosperm may either completely consumed by the developing embryo, example in groundnut and beans before the maturation, or it persists in the mature seed. For example, castor and coconut are used up during the seed germination. ಕೆಲವೊಂದು ಸೀಡ್ಸ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಎಂಡೋಸ್ಪರ್ಮ್ ಕಂಪ್ಲೀಟ್ ಆಗಿ ಕನ್ಸ್ಯೂಮ್ ಆಗಿಬಿಡುತ್ತೆ ಎಂಬ್ರಿಯೋದಿಂದ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ವೇರ್ ದ ಎಂಡೋಸ್ಪರ್ಮ್ ಇಸ್ ನಾಟ್ ಪ್ರೆಸೆಂಟ್, ಗ್ರೌಂಡ್ನಟ್ ಅಂಡ್ ದ ಬೀನ್ಸ್. ಬಟ್ ಇನ್ ಸಮ್ ಸೀಡ್ಸ್ the endosperm is remaining, so that is used for the germinating the seed. Okay, for example, coconut and the castor. So that is the example. ಇದನ್ನ MCQ ದಲ್ಲಿ ಕೇಳ್ತಾರೆ. Endosperm is remaining in which of the following? So that is castor and the coconut. And endosperm is completely consumed by the embryo. So which of the following? Pea, groundnut, beans. So these are the examples. Then embryo. So the zygote, where the male and female gametes are fused, that will develop into a embryo. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. So it divides continuously. Nodi zygote enagate completely, it will undergo the mitotic division and it will develop into a globular embryo stage, heart shaped embryo stage, and the mature embryo stage. Okay, so this embryo that will divide and become globular embryo and heart shaped embryo, and that will become mature embryo. These developments that is similar in the monocots and the dicots. Okay, monocot matte dicot alli embryo development enide that is same, but the mature embryo that is different in dicot and the monocot. The structure of the dicot embryo, dicot plant alli embryo yav tara irutte? A typical dicotyledonous embryo consists of embryonal axis and two cotyledons. So these are the two cotyledons. The portion of embryonal axis above the level of cotyledons is called as the epicotyle, which terminates with the plumule or the stem tip. So here, so this is the epicotyle, and epicotyle ends in the region of plumule. Plumule, that is also known as the stem tip. The cylindrical portion below the level of cotyledons is the hypocotyle. So this is the hypocotyle region, you can see here, and that terminates at the lower end in the radicle. Here the hypocotyle end, so that ends here at the radicle, and the root tip is covered with the root cap. So this radicle, that is called as a root tip, and plumule, that is the shoot tip, that will develop, plumule develop into a shoot and the radicle develop into a root. Here it is covered by the root cap. ಇಲ್ಲಿ diagram ಮತ್ತೆ ನಿಮಗೆ explanation ಅನ್ನ ಕೇಳ್ತಾರೆ. And what is the structure of the monocot embryo? So this diagram is also very important. Monocot embryo diagram. Embryos of monocotyledonous possess only one cotyledon. In the grass family, the cotyledon is called as a scutellum. So this is the single cotyledon, which is situated towards one side of the embryonal axis, and its lower end, the embryonal axis has the radicle. Its lower end which has the radicle, and root cap enclosed in an undifferentiated sheath, that is the coleorhiza. Here this is the root cap that is covered with the structure coleorhiza. The portion of embryonal axis above the level of attachment of the scutellum, that is the epicotyle. Epicotyle has shoot apex and leaf primordia enclosed in a hollow folia structure, that is the coleoptile. Here this shoot apex that is covered by a structure, that is the coleoptile. So that is the structure of the monocot embryo. And the embryo, the seed, the ovule develop into a seed, right? In angiosperms, the seed is the final product of sexual reproduction. So the ovule, that is develop into a seed and seeds are formed inside the fruits. A seed typically consists of seed coat, cotyledons and the embryonal axis. The cotyledons of embryo is simple structure, generally thick, swollen due to the storage of the food reserve. So here mature seeds, there is two types of seeds, albuminous seeds and the non-albuminous seeds. What you mean by non-albuminous seeds? Very important. So they do not have residual endosperm as it is completely consumed by the embryo development. Already we have studied pea and the groundnut. Albuminous seeds means they retain the part of the endosperm which is not completely used up during the embryo development, that is wheat, maize, barley and the castor. So that these are the non-albuminous seeds. And in some seeds, the black pepper and beet, these all examples are very important. Remnants of nucellus are also persistent. So the nucellus is also remains, that is known as a perisperm. What is perisperm? That is also very important question. What is perisperm? And examples. So which of the following contain the remnant of the nucellus? Black pepper and beet. Very important. So here you can see the dicot seed and this is the structure of the monocot seed. In dicot seed, cotyledons and micropylar end. And in this seed you can see the endosperm is remaining here. Here, the integuments of the ovules harden as tough protective seed coats. The integuments of the ovules, they will become seed coat and the micropyle remains as the small pore in the seed coat. A small pore ತರ ಇರುತ್ತೆ micropylar region ಏನಿದೆ ಅಲ್ವಾ, ನೀವು dicot seed ಅನ್ನು ನೋಡಿದ್ರೆ ನಿಮ್ಗೆ ಕಾಣಿಸುತ್ತೆ, that is the micropyle region. This facilitates the entry of oxygen and water into the seed during the germination. As the seed matures, its water content reduced and seeds become relatively dry. The general metabolic activity of the embryo slows down. When the seed become mature, the metabolic activity of the embryo which is present inside the seed, the embryo may enter the state of inactivity called dormancy or favorable conditions are available. So when the when the favorable conditions are required, then embryo will develop into a that is able to germinate. The ovules mature into seeds, the ovary develops into fruit. The transformation of the ovules into seeds and ovary into fruit proceeds simultaneously. The wall of the ovary develops into wall of the fruit called pericarp. The wall of the ovary, that is called the fruit wall, that is the pericarp anta karitare. The fruits may be fleshy in guava and orange, mango, fleshy fruits, or sometimes dry fruits also there, groundnut, mustard, and many fruits have evolved mechanism for dispersal of the seeds. Then you know that ovary develops into a fruit, but few species like apple, strawberry, cashew, so the thalamus also contributes to the fruit formation. Such fruits are called as a false fruits. So what are false fruits? Examples for the false fruits anta keltare. The ovary, you know that ovary develops into a fruit. This develop into a fruit, but sometimes the thalamus part is also contribute for the fruit formation. Such type of the fruits are called as the false fruits anta karitare. Iga apple irbodu or cashew, strawberry. So these all are example for the false fruits. And some fruits, they develop without fertilization. They are called as parthenocarpic fruits. These all definitions are very important. So ಕೆಲವೊಂದು fruits ಏನಾಗುತ್ತೆ, so they develop without fertilization. They are called as parthenocarpic fruits. For example, banana. So parthenocarpy can be induced through application of growth hormones, and in such fruits are seedless fruits. Parthenocarpic fruits, they formed without fertilization and they may be seedless. Example, banana. Okay. In few species, the seed lose the viability within a few months. Seeds of large number of species live for the several years. So ಈಗ seeds ಅಲ್ಲಿ ಕೆಲವೊಂದ್ಸಲ ಏನಾಗುತ್ತೆ, so they remain their viability. So they remain they alive around hundreds of years. Seeds ಏನಿರುತ್ತೆ thousand hundreds of years ಇರ್ಬೋದು. For example, the oldest is the lupine, that is Lupinus arcticus. So that is available, that is found in the Arctic tundra region. Very important, Lupinus arcticus. And the seed germinated and flowered after estimated record of 10,000 years of dormancy. So ಕೆಲವೊಂದು ಸೀಡ್ಸ್ ನೋಡಿ 10,000 years dormancy ಆಗಿ ನೆಕ್ಸ್ಟ್ ಅದು ಫ್ಲವರ್ ಅನ್ನ ಫಾರ್ಮೇಶನ್ ಮಾಡಿದೆ. A recent record of 2,000 years old viable seed of date palm, that is Phoenix dactylifera, discovered during the archaeological excavation at the King Herod's palace near the Dead Sea. So these all examples are very important. So the last topic of this chapter, that is Apomixis and polyembryony. So although seeds in general are products of the fertilization, seeds ಹೇಗೆ ಫಾರ್ಮೇಶನ್ ಆಗುತ್ತೆ, ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗಿ ಮೇಲ್ ಅಂಡ್ ಫೀಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಫ್ಯೂಸ್ ಆಗಿ ನೆಕ್ಸ್ಟ್ ಫಾರ್ಮೇಶನ್ ಆಗುತ್ತೆ. ಬಟ್ ಫ್ಲವರಿಂಗ್ ಪ್ಲಾಂಟ್ಸ್ ಸಚ್ ಆಸ್ ಸ್ಪೀಶೀಸ್ ಆಸ್ಟರೇಸಿಯೆ, ನೋಡಿ ಇಂಪಾರ್ಟೆಂಟ್ ಇದೆ, ಆಸ್ಟರೇಸಿಯೆ and grasses have evolved the special mechanism to produce the seeds without fertilization called apomixis. What is apomixis? The seeds are produced without fertilization. So ಎಲ್ಲಿ ನಾವು ಅದನ್ನ ನೋಡಬಹುದು, ಆಸ್ಟರೇಸಿಯಸ್ ಅಲ್ಲಿ ನೋಡಬಹುದು, ಆಸ್ಟರೇಸಿಯೆ ಅಂಡ್ ಗ್ರಾಸಸ್ ಅಲ್ಲಿ ನೋಡಬಹುದು. Okay, there are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization. So that is the apomixis. And what is polyembryony? In many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac start dividing, protrude into a embryo sac and develop into a embryos. Here, more than one embryos are formed. So that is called as a polyembryony. In such species, each ovule contains many embryos. Within a ovule, one embryo irutte alva, fertilization agi develop agirodu one embryo, but the embryo, so that is not one embryo within the ovule, many embryos are present. So that is called as a polyembryony. Occurrence of more than one embryo in a seed, that is referred as the polyembryony. Here, ಹೇಗೆ ಪಾಲಿಎಂಬ್ರಿಯೋನಿ ಆಗುತ್ತೆ, ಇಲ್ಲಿ ನೀವು ಸಿಟ್ರಸ್ ಅಲ್ಲಿ ಮ್ಯಾಂಗೋ ವೆರೈಟೀಸ್ ಅಲ್ಲಿ ನೋಡಬಹುದು. ಸೊ ದಟ್ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಸ್ಟಾರ್ಟ್ ಡಿವೈಡಿಂಗ್. ಸೊ ಅದೇನಾಗ್ಬಿಡುತ್ತೆ, ನ್ಯೂಸೆಲ್ಲಾರ್ ಸೆಲ್ಸ್ ಸರೌಂಡಿಂಗ್ ದ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಸ್ಟಾರ್ಟ್ ಡಿವೈಡಿಂಗ್. ಸೊ ಡಿವೈಡ್ ಆದಾಗ ಏನಾಗುತ್ತೆ, ಸೊ ದಟ್ ಡೆವಲಪ್ ಇನ್ ಟು ಅ ಮೆನಿ ಎಂಬ್ರಿಯೋಸ್ ಡೆವಲಪ್ ಆಗುತ್ತೆ. ಸೊ ದ ಡೆಫಿನೇಷನ್ ಇಸ್ and examples. So these are very important. What is polyembryony? Give example. How the polyembryony is formed? Examples. So these all are very important for your exam. Okay. And here, so this is the last topic of this chapter, sexual reproduction in the flowering plants. Okay. So this is the just brief explanation. ನಾನು ತುಂಬಾ ಡೀಟೇಲ್ ಆಗಿ ನಿಮಗೆ ಹೇಳಿಲ್ಲ, ಬಟ್ so that is very important for your exam, brief explanation for your CET. Okay, so ಇಲ್ಲಿ ನಿಮಗೆ ನೋಟ್ಸ್ ಎಲ್ಲ ಸಿಕ್ಕಿದೆ ಅಂತ ಅನ್ಕೊಳ್ತೇನೆ. ಸೊ ಅದರಲ್ಲಿ ನಿಮಗೆ ಇಂಪಾರ್ಟೆಂಟ್ ಟಾಪಿಕ್ ಅನ್ನ ನೋಟ್ ಮಾಡ್ಕೊಂಡು ಓದ್ಕೊಳ್ಳಿ. ಇಫ್ ಯು ಹ್ಯಾವ್ ಎನಿ ಡೌಟ್ಸ್ ಯು ಕ್ಯಾನ್ ಆಸ್ಕ್. Okay. Yes, thank you dear students. We'll meet in the next session.